everyone, and welcome back to PodBites, where we break down complex medical topics into bite-sized pieces, perfect for your commute, coffee break, or whenever your curiosity calls. I'm Naem, your host for today's episode, and joining me is our amazing speaker, Samhar.
Hi everyone. Today we are diving into a topic that shows up in biochem, physiology, pathology, and every exam question that wants to ruin your day. Carbohydrates and glycosaminoglycans. Buckle up, this one basically a full buffet.
This is a big one, but don't worry, we're breaking it down in a way that actually makes sense. So grab your notes, or just relax and listen. Let's get into it. Okay, Samhar, so we always hear that carbs do a lot of things. Some people say that it's the energy source, the thing that gives us power. So can you actually give us a real breakdown beyond the energy source answer?
Definitely. Carbohydrates do a lot more than giving energy. They act as fuel molecules like glucose and fructose. They are also storage molecules like glycogen and starch, basically the body's power banks. Also, carbohydrates forms important structural components like ribose in RNA and deoxyribose in DNA. They participate in cell communication through glycoproteins and glycolipids, and they serve as recognition sites. For example, viruses bind to cell surface glycoproteins, toxin binds to a specific glycolipid, and H. pylori attached to a gastric glycoproteins. They also help with immune signaling, like selectins. They help neutrophils bind to endothelium. And an important one for pathology, the mannose 6-phosphate receptor in the Golgi apparatus. It tags lysosomal enzymes and sends them to the lysosome.
Okay, so wait, the last part was kind of huge. So the mannose 6-phosphate receptor is like a shipping label to the enzyme. It tells the enzyme, go to the lysosome. Not the ER, not the mitochondria, the lysosome.
Exactly, and when that fails, we get cell disease. More on that later.
Okay, Samar, so let's go a little bit deeper, shall we? So when we say monosaccharides, what are the categories that actually we need to know?
We classify monosaccharides based on the carbonyl group they have. If they have aldehyde group, then they are aldoses. If they have ketone group, then they are ketoses. We also name them by the number of carbons they have: triose, pentose, hexose, and so on. Then come the isomers. First, we have structural isomers. They have same formula, different arrangement. Then we have stereoisomers. Same formula, different 3D shape. Also, we have enantiomers, which have mirror images. And finally, we have epimers. They differ at exactly one carbon. A classic example for that is glucose and galactose. They differ only at carbon number 4.
Ah yes, glucose and galactose, the famous C4 epimers. That tiny difference that makes lactose intolerance possible. So somehow let's dive more into talking about sugars. So textbooks would draw sugar in a straight sticks, but in real life there are rings, right? Why is that?
Because in solutions, the carbonyl group reacts with a hydroxyl group, forming either pyranose ring, which is 6-membered, or furanose ring, which is 5-membered. This reaction creates the anomeric carbon, giving rise to the alpha and beta forms of anomers.
And that tiny alpha and beta difference is why we can digest starch, but not cellulose. That always blows my mind. Okay, Samhar, let's talk about reducing sugar. I swear I memorized the definition once, but could you actually re-explain it in a way that sticks this time?
Sure thing. A reducing sugar is any sugar with free anomeric carbon that can donate electrons. These sugars reduce Benedict's reagents. Most monosaccharides and most disaccharides are reducing, except sucrose, which is the common non-reducing exception.
Earlier you brought up Benedict's test. Can you please break it down for me? What exactly is it, and what does it actually tell us?
Sure thing. Benedict's test is used to check for reducing sugars, such as glucose, fructose, lactose, and maltose. The reagent starts out blue because it contains copper ions. When a reducing sugar is heated with Benedict's solution, it donates the electron to these copper ions. As the copper ions are reduced, the blue color fades and new solid particles are formed. These appear as green, yellow, orange, or brick red, depending on how much reducing sugar is present.
You mentioned that the colors change, but I don't really remember what each color means. Can you please emphasize on that?
The color change correlates with how much reducing sugar is present. Green is about 0.5%, yellow is about 1%, orange is about 1.5%, and finally, brick red is about 2% or more.
Okay, I think I get it now. So Benedict test is not just a positive-negative type of test.
Nope, it's semi-quantitative.
Somehow we always hear that high blood sugar can damage protein. But what's actually happening there? I still don't understand how can sugar damage protein. How are they related? What kind of harm they cause?
Great question. Glucose can bind to proteins non-enzymatically and forms AGEs, or what's called advanced glycation end products. These AGEs accumulate over time, damage proteins, and contribute to diabetes complications.
So high sugar literally caramelizes the body proteins over time? That is terrifying.
That's also how hemoglobin A1C forms. Glucose binds to hemoglobin, and because red blood cells live about 120 days, hemoglobin A1C reflects long-term glucose control for about 3 months.
So basically you're saying it's a test to expose the patient. We know exactly what they have been doing for the past 3 months.
Exactly, no secrets.
So oligosaccharides always felt like not the most important content, it's always thrown at the last couple of slides in each PowerPoint. But apparently, they're very important.
They are important. Oligosaccharides have 2 to 10 monosaccharides units. They are partially digested by gut bacteria and contribute to gut health. They increase intestinal viscosity, slow glucose absorption, increase satiety, and reduce the risk of obesity and type 2 diabetes. They are linked by glycosidic bonds formed through condensation reaction, which releases water.
So Samhar, let's talk a little bit more about polysaccharides. We always hear about amylose and amylopectin. And before you start talking, let me make a guess. Amylose is the linear one, and amylopectin is the branched one.
That's correct. Amylose is a linear polymer with alpha-1 4 bonds because it's tighter and more compact. It's digested slowly and causes smaller glucose spikes. A simple mnemonic for that is amylose is slow. You get it? Amylose, slow. Then we have amylopectin. Amylopectin is branched, containing both alpha-1 4 and alpha-1 6 bonds. It's digested faster and causes higher glucose spikes. A mnemonic for that is amylopectin, plenty of branches.
So you're telling me right now when I'm sleepy after eating my rice meals, it's that goddamn amylopectin doing it.
Yeah, exactly.
So we talked about amylose and amylopectin, but what about glycogen? How is it actually different from all those?
Glycogen look like amylopectin, but with even more frequent branching. It's ideal for rapid glucose release, muscle use it for quick energy, and the liver uses it to maintain blood glucose levels.
Okay, so let's move to another segment. This is the part that actually always scares me. Can we start it simple? What in God's grace is glycosaminoglycans?
Well, glycosaminoglycans, or also called GAGs, are long unbranched heteropolysaccharides made of repeating disaccharides units. One amino sugar, like gluconac or galnac, and one acidic sugar, like glucuronate or iduronate. They carry a strong negative charge which attracts sodium and water. This gives tissue hydration, viscosity, and resistance to compression, like synovial fluid in joints.
So you're telling me the negative charges not only pulls water in, they also repel each other, creating that slippery extracellular matrix texture.
Exactly, you've got it.
Okay, Samhar, so this is the part where people usually get overwhelmed. What are the different types of GAGs? Can you walk us through the main ones that we actually need to know as med students?
For sure, there are 6 main GAGs to know. Hyaluronic acid, which is unsulfated and not attached to a protein, chondroitin sulfate, which is the most abundant, dermatan sulfate, which contains hyaluronic acid, keratin sulfate, which is unique because it has no uronic acid, heparin sulfate, which is found in cell surface and basement membranes, and finally, heparin, which is highly sulfated, produced by mast cells, and acts as an anticoagulant. And don't worry, you don't need to memorize their chemical structures.
Thank you for telling us not to memorize their structures, because I assure you, I wouldn't have been able to. And somehow I always hear about hyaluronic acid being the odd one. What makes it different from the other GAGs?
Yeah, hyaluronic acid is definitely the special case. Its sugars are synthesized in the cytoplasm, then assembled right at the plasma membrane and secreted directly. It doesn't go through any Golgi modifications, and it's not attached to a core protein.
Wait. Before we go forward, what is a core protein?
Proteoglycans have a core protein with GAG chain attached to a tetrasaccharide linker, xylose, galactose, or glucuronic acid. They help organize the extracellular matrix and influence signaling. Heparin sulfate also anchors lipoprotein lipase, allowing muscle cells to access fatty acids from lipoproteins, which is pretty efficient if you think about it.
So let's talk a little bit about the degradation of GAGs. Correct me if I'm wrong, but that happens inside the lysosome, doesn't it?
Yup, GAGs are degraded in lysosomes using glycosidase and sulfatase. If one enzyme is missing, partially degraded GAGs accumulate, causing mucopolysaccharidosis, or MPS disorders. These conditions causes hepatosplenomegaly, skeletal changes, coarse facial features, corneal clouding in some types, developmental delays, and GAG fragments in the urine.
Okay, Samhar, when it comes to mucopolysaccharidosis, the names can get confusing. What are the key differences between Hurler, Chi, and Hunter syndrome?
Great question. Hurler syndrome is the classic severe form, and it does have corneal clouding. Chi syndrome is much milder version of the same enzyme deficiency. On the other hand, Hunter syndrome is different. It's X-linked and it has no corneal clouding. The mnemonic I like to use is Hunter needs clear vision.
So what you're telling me that a hunter is a boy, that's why it's X-linked, and he needs to see, that's why there's no corneal clouding.
Yup, that's it. We also mentioned ISIL disease earlier. In ISIL disease, enzymes fail to get the mannose-6-phosphate tag in Golgi. Without the tag, they never reach the lysosome and end up being secreted outside the cell.
So the lysosomes becomes like an empty room with the trash piling outside, basically. Alright, let's test what we have learned in a quick case. A child presents with developmental delay, coarse facial features, corneal clouding, and dermatan sulfate detected in the urine. What's the diagnosis that should immediately come to mind?
That constellation of findings is classic for Hurler syndrome. Honestly, the case is practically shouting it.
Okay, Samhar, so it was a big session today. Can you please at least give us a quick summary about what we have covered for today?
Sure thing. Today, we covered everything from carbohydrate basics, reducing sugars and polysaccharides all the way to GAGs, proteoglycans, lysosomal degradations, ISIL disease, and the mucopolysaccharides. If you understood this episode, you have basically mastered the entire lecture, and honestly, that's a great feeling.
This was one of the most complete and clearest breakdowns I've ever gone through. If biochem always sounded like this, I assure you my grades would have looked better. Thank you, Samhar, for joining us today. Thank you guys for listening. Stay curious, stay consistent, and we will see you next time on PodBites. Take care.